Okay. Well, good morning, everyone. Thanks so much for joining us and being here for today's presentation. My name is Amy Jewitt, and I am the Invasive Species Coordinator at the Western Pennsylvania Conservancy and the Pennsylvania Natural Heritage Program. And I'm joining you today from my home office in Valencia, Pennsylvania, and happy to have everyone on today. We are expecting a little over 100 people. So right now we have about 40 people online. So thanks for being here. I do want to mention as far as today's presentation and any questions that you might have, there is a Q&A feature that is available for folks if you would like to type in a question during today's presentation. But we will be taking some time at the end to go through and ask and have those questions asked to our presenters. So you can type in during or after, and we will have time at the end to answer questions. So today's presentation is called 4 Questions to Ask When Working with Freshwater Snails: Who, How, Why, and What Now? And we have 3 presenters that are with us today from Penn State Behrend. And I want to take just a moment and introduce each of our 3 presenters. So first up, we have Dr. Lynne Beaty. Lynne is an assistant professor of biology at Penn State Erie, the Behrend College. As a professor and behavioral ecologist, Lynne teaches undergraduate classes on ecology, conservation biology, animal behavior, and experimental design, and conducts research on the behavior and population dynamics of native and invasive snails, salamanders, and frogs. And then next we have Dr. Sam Nutile. Sam is an assistant professor of biology at Penn State Erie, the Behrend College. At Behrend, Sam teaches undergraduate classes in introductory biology, aquatic ecology, ecotoxicology, and science communication. Sam's research focuses on the effects of invasive species and anthropogenic chemicals in aquatic ecosystems. And then finally, we also have with us today Dr. Adam Simpson. Adam is an assistant teaching professor of biology at Penn State Erie, the Behrend College. Adam teaches undergraduate classes in anatomy and physiology, ecology, and ecotoxicology. As an environmental physiologist, his research focuses on the physiological adaptations that enable native and invasive animals to survive in changing environments. So thanks to all 3 of you for being here today. And without further ado, I will turn it over to you, Lynne.
Right. Thanks for that introduction, Amy. We're so glad to be here. So we're going to talk to you guys, or take turns talking to you guys today about aquatic invasive snails. So we'll just kind of rotate out as we get to different sections. Like Amy said, my name's Lynne Beaty, and I'm going to be talking to you guys first. So one of the issues, as I'm sure many of you are aware with managing invasive species, is knowing who you have and knowing where they are. Because it's particularly difficult to control or even understand the invasive species that you're working with if you don't know what's there. And this can be pretty problematic for some of our less charismatic species, like some people consider snails to be less charismatic. Their identity is a lot less well understood than some more commonplace invasive species. And some of the issues with identifying and managing invasive snails too is that their effects aren't necessarily overt. You don't see them, often you don't see them taking over landscapes. Some of their effects tend to be more subtle, where they're competing with native species of snails, which many people might not know how to distinguish between native and invasive individuals anyway, or they might be doing things like altering parasite transmission, which you might not notice unless there's a massive die-off. So what we're going to do today is talk to you guys about 4 different aspects of aquatic invasive snails. First, introducing you to who some of our major players are in Pennsylvania as far as aquatic invasive snails. We'll talk about how you can look for these guys if you are interested in conducting your own invasive snail surveys, and how you can report those, why you might be interested, and why we're doing what we're doing. So we'll tell you guys a little bit about some of the research that we have going on, and then what you guys can do in the meantime or on your own time. So I'm going to talk to you guys about the who of aquatic invasive snails. And I think before I get started telling you guys specific species to look for, I think we need to go over snail anatomy a little bit, because there are certain terms that I'll be mentioning when I go through the different invasive species that you'll need to keep in mind. And I just want to make sure everyone's kind of familiar with all of these terms before I go forward. So we'll start with a generic snail shell. Each of these kind of main tubes of the snail are called whorls. Snails, as they grow, start up top here, and then they grow downward. They do change direction, though, depending on the species. There are some species of snails that grow clockwise. These are dextral snails, so their opening is going to be on the right-hand side. And there are some species that grow the other direction. Their openings on the left-hand side. These are called sinestral. So if you pick up a snail and you're looking at the opening of the snail, the opening's on the left-hand side, that's a sinestral snail. It's on the right-hand side, that's a dextral snail. So of our whorls, they're usually divided into two main categories. The biggest whorl at the bottom here is your body whorl. That's where most of the tissue of the snail is. That's where most of their organs are present. All of the other whorls above that are considered the spire. So that shape of the spire and the relative size of the spire relative to the body whorl can be important in distinguishing different species, as well as the structure of that spire, how angular it is, whether or not it's compressed. That can be pretty important trait to look at. Other key traits to look at when you're trying to identify snails are the aperture. So the aperture is just another name for the opening that those snails have. The outside edge of the aperture is called the outer lip. The inside edge is called the inner lip. There's also, and you can perhaps see it on this snail here, this area, this little indent here, it's called the umbilicus. I like to think of it as like the snail's belly button. That's where the snail kind of grows around, and you can't always see it in all species, and that's going to be one of our identifying features, is whether or not you can see that umbilicus, how obvious it is, or if it's just like a little slit. Another characteristic that some snails have is called the operculum. So instead of just seeing snail foot here when you look at the snail, whenever you look at the aperture, you might see that there is essentially a trap door in place for that snail. And obviously this serves a number of protective functions for that snail, as they can close themselves in entirely to protect themselves from predation and things like that. But not all snails have opercula. That's one way that you can tell some native species from some invasive species. And the growth patterns on that operculum can be quite important for distinguishing different species. So there are four main growth patterns of this trapdoor. So again, not all snails are going to have this trapdoor. Those that have it have certain growth patterns in it, because as that snail continues to grow along those whorls, this trapdoor either grows outward or kind of forms rings. So if you look at one of these trapdoors and it looks like there's like a cross section of a tree, it has concentric rings on it. This is called a concentric pattern to the operculum. So you just have kind of non-overlapping rings of growth on that trapdoor. Sometimes snails have some of that concentric growth pattern, but if you look at the very middle, it's a spiral. So that means when that snail was really small, it has a spiral pattern to that operculum, and then as it grows, it forms these concentric rings. Another type of pattern for the operculum is called paucispiral. So in this pattern, it looks almost like a nautilus, where it starts as a spiral, and then it gets large quite fast. So you won't see too many outside rings as you get more towards the outer lip portion of that aperture. The last type is called multispiral. So in the multispiral, looks like a spiral that goes round and round and round and round. And you can kind of tell that there's going to be differences in aperture shape for each of these different types of operculum as well. So snails that have multispiral opercula tend to have more circular apertures or openings than those that have concentric or paucispiral apertures. So I also mentioned the umbilicus as a trait to identify or distinguish between different species of snails. And there's really three main shapes it can take. So one is called the imperforate umbilicus. So in this case, you can't see an opening for that belly button, that umbilicus at all. A perforate shell has a little bit of an opening for that umbilicus. You can tell that it's there, but it's not particularly obvious. And then there are some snails that we just call umbilicate shells because that umbilicus is very obvious. You can almost see up through the top underside of the spire when you look up through that belly button there. All right, so let me get to some of our main invasive snail species that we have here in Pennsylvania. So for each of these species, what I'm going to do is I'm going to give you the key ID features for that species. I'll mention some of its documented or plausible impacts on native ecosystems, and then mention some of its native lookalikes that might be confused for these invasive species. And I will say some of these invasive species are really difficult to identify. So when in doubt, I would always seek out expert identification for them because some of them are really difficult. So the first species I have here is called the Mudbithynia, the faucet snail. These snails are not particularly large. They range from 6 to 15 millimeters in length. So you're looking at something that is a centimeter, centimeter and a half. Most of the individuals in the Great Lakes are only about 12 millimeters in length. So pinky nail-ish in size, quite small. They're usually, if they are living, they have this pale brown shell. Once they die, they do turn white, as most snail shells do. For their body size, they do have quite a large spire. So again, if we look at this body whorl and then compare the relative size of that spire to the body whorl, that's a pretty big spire for something this small. They also have an imperforate umbilicus, so you can't quite see it in this angle, but if you were to look, or you look on the other side, you wouldn't see an umbilicus because there isn't one, or it's covered up in that area of the shell. They also have an operculum that is concentric with a spiral center. So you'll see that little spiral in the middle, and then we see almost like tree growth rings on the outside. And one unique feature of this snail, which other native snails do not have, is this operculum is calcareous. So most of our native snails have a corneous operculum, while this one is actually made of calcium carbonate. So their operculum is a lot harder than native snails. A lot of our native snails have a fairly flexible operculum because it's made out of proteins instead of calcium carbonate. So these guys, where they are present, can form pretty high densities. They are known to outcompete native snail species in some areas, or at least result in a reduced diversity of native snail species in some places. And they are known to transmit parasites to a number of wildlife. There was, I believe, an instance of waterfowl die-off from snails, or from waterfowl consuming these snails, which contained an abundance of parasites. But a lot of effects of these snails are not known. So you'll notice for a lot of the snails I'm going to mention, these effects are going to generally be the same. And then I have a question mark here because there's a lot that we don't know about the impacts of these snails. So there are a couple of native species that look similar to the faucet snail, the Mudbithynia. I have some of the more tricky ones posted here. So the first one I have is a physid. This one you can pretty easily distinguish from the Mudbithynia because it's sinestral, where our faucet snail is dextral. Remember, sinestral, the opening is on the left-hand side. Dextral, it's on the right-hand side. They also lack an operculum. Linneids, there's a couple of species in here that could look fairly similar. These guys are generally bigger than faucet snails will be. And you'll notice that the spire isn't quite as large. These guys also do not have an operculum. So if you see a snail without an operculum, it is not a faucet snail. Our last little guy over here, M. Nicola lamosa, does have an operculum, but it is paucispiral. So it's that opening spiral coming out, and they are much smaller. And their operculum is corneous. It's made of protein instead of calcium. So in Pennsylvania, these guys are primarily reported in Lake Erie, or in the Lake Erie watershed. There aren't too many reports elsewhere in the state. Doesn't mean that they're not elsewhere in the state. I think for a lot of these maps that I'll show you, odds are that these snails are located at a number of other locations. They just haven't been reported yet. So keep an eye out for these guys. They could be near you. All right, second species I have here is called the big-eared radix. You can probably tell why it's called the big-eared radix because it looks like a big old ear. Although this photo is a little misleading because the snail that you're looking for is actually about 3.5 centimeters large, so it's not that huge, but it does look like an ear. In life, these snails are kind of yellowish tan. They're pretty easily distinguished by this huge aperture and really tiny spire. So they have this bulbous body whorl here, tiny spire, huge aperture. That aperture is almost 50% or greater than 50% of their total length, which is unlike any other snail that we have here. Their shell is really thin and they do not have an operculum. So again, you'll note I have the same impacts generally here for these guys. We know they do compete with native snail species. They are closely related to limnaeids. So they look like, oops, my apologies. They look like a number of other limnaeids, but they do have this fairly large body whorl and really big aperture compared to some of the native ones, although they do compete with those guys that look fairly similar, and they are known to transmit parasites. These guys are primarily documented in the eastern part of the state. Again, just because they're only documented there doesn't mean they aren't elsewhere. These snails can be, all of the snails can really be transmitted quite easily or transported quite easily. So keep an eye out. All right, our third species here is the European stream valvata. And in case you guys haven't noticed the scale bar that I have here, these snails are very small. You're looking at 0.5 centimeter up to 0.7 of a centimeter or so. So these guys are really little, very easily missed, very easy to transport. In life, these guys are yellowish and they have spots from the underlying tissue underneath. They also have this really weird external respiratory organ. So here you can see that yellowish of the shell. And these two structures here are the respiratory organs. So they have one, it's kind of this long stalk-like portion, and then the other looks more like a traditional gill. This is going to tell you that it's one of these guys. At least belongs to this group, is this weird external respiratory organ. Their shell can be variable in height. It kind of depends on the environmental conditions that they're in, where if they're in more eutrophic conditions, they tend to grow a bit taller compared to other conditions. And they have one of those multi-spiral operculum. You can see that pattern pretty well, where it just looks like one of those spirals constantly going out. These guys do also compete with native species. There are other valvatas around they compete with, and they are known to transmit parasites. As far as our native lookalikes, there are two that look very similar to this particular snail. This is one that I would definitely recommend having someone double check the ID for. The first is our mossy valvata. The second is the fringed valvata. So the main difference in these guys, and I have a side-by-side comparison here, where this diagram is the invasive shell. This is your valvata, and this is your, sorry, that's your mossy one, and this is your fringed down here. So the key things or the key distinguishing features between these species are the umbilicus. So in our invasive individual, the umbilicus is a lot smaller than our native two snails. The other two are almost umbilicate, where it looks like you can see the top of the spire from the inside. And if we look at spire height and spire angle. So in our invasive individual, it's more the rings of the body, sorry, the whorls are on top of each other, and it's fairly tall. Whereas in our native two species, it tends to be a much shorter spire, and it almost is at an angle. The fringed valvata as well has those fringes on it, which are going to make it look quite different. These guys right now have not been reported, at least definitively reported in Pennsylvania, but they are in nearby water bodies. So like they've been reported in other bodies of, or in other portions of Lake Erie. So odds are they're along this Lake Erie coast. They've also been reported the border of New York. So odds are that they're probably on this side of the state as well. Another species, the New Zealand mud snail. These guys are also quite small. If you look at our scale bar here, 4 to 6 millimeters, but they're often a lot smaller than that. Most of the populations that we have here are female only. They're parthenogenic lines, so they can produce themselves without males present. They're usually this brownish grayish color. In some populations, you'll see this keel on the whorl, but not always. So that's not going to be a definitive characteristic for them. They do have this deep furrow between the whorls, which makes it look to me like a unicorn horn compared to some other species of snail. And they have a really thin paucispiral operculum. So that's that nautilus outward operculum pattern. These guys get incredibly dense. Fouling is one of the main impacts that they have. They can be upwards of 300,000 individuals per meter squared, wherever they have really taken off, owing in part to the fact that, they're parthenogenic, they don't need males. But given how dense their populations can be, they have the opportunity to have quite an impact on native ecosystems, including competing with native snail species. They can also alter nitrogen and phosphorus regimes just by sheer abundance. There are also a number of native species that look like New Zealand mud snails. These are incredibly difficult to tell apart. For some of these species, it comes down to differences in penile structure of the snail. So in this case, you definitely want to consult with an expert before definitively reporting that this is them. I know for this last species I have over here, they tend to be a little bit more terrestrial than New Zealand mud snails. So that habitat difference will probably be a good indicator to you, but the other ones are going to be quite similar. So these guys have been reported in a number of locations in Pennsylvania. And odds are they're probably a number of other places, given their size and given how quickly and fast their populations can increase. All right. Another species is the banded mystery snail. So the banded mystery snail is an odd one because it's actually a native transplant. It's not native to this area, but it is from the other parts of the US. These guys are moderately sized, so they do get up to almost 5 centimeters in length. Most of them are a bit smaller than that, though. And they're called the banded mystery snail because they have bands on them, although sometimes they don't. That's one thing to keep an eye out for, although most of the populations that we're aware of have bands on them. It is possible that it does not have bands. They still usually have about 3 to 5 whorls in total. They're pretty deep grooves between them. Some people call this a shouldered whorl because there's a pretty strong horizontal component to that whorl after that suture in the middle there. And they have a concentric operculum, so tree growth rings. These guys are known to compete with native species and transmit parasites. They also have there's some evidence that they reduce the survival of largemouth eggs. That comes from a study in the 70s, though I haven't seen a follow-up to that study, but it is possible that they can impact fish egg hatching success. There are some species that look like the banded mystery snail. One is this pointed campyloma here. You can kind of see, though, that the body whorl isn't nearly as bulbous as this banded mystery snail. These guys also do not have bands. So if you see bands, it's definitely a banded mystery snail. If it doesn't, double check the shape. And this Leoplax has quite a bit of texture on the shell. So you would notice that as a distinguishing feature. As of right now, these guys have only been reported in one part of Pennsylvania, although I have a report to add for up over here. We found one this summer in Lake Canadota. So it was just one, but I'm working on it, Amy. We're going to get that reported. All right. So for our last set of invasive species to introduce you to, I have the Asian mystery snails. And as of right now, there are 2 species reported in the US, the Chinese and the Japanese mystery snail. I have both of them on the same slide because while they can sometimes be distinguished morphologically, there are instances where you cannot use morphology to distinguish between these 2 species. You can tell if it is one of these two primarily based on size. These snails are big snails. So a lot of times the Chinese mystery snails are upwards of 5 centimeters in length, and the Japanese can get upwards of 7. All of our native snails are not nearly that big. So if you see a giant snail, could be one of these guys. They do have a lot of whorls of growth. They live for a particularly long time. These are the species that we study, so these are the ones that we know the best. But like I mentioned, they're not always able to be distinguished from each other morphologically, because this Japanese mystery snail has a morph that looks exactly like the Chinese mystery snail. So very difficult to identify between these two, but they do stand out from native species based on size. Their operculum, much like that other banded mystery snail, is a concentric corneous operculum. We know that they compete with native species. They've been associated with parasite spreads. They also bioaccumulate a whole bunch of metals, which can be good and bad. From removing it from the environment perspective, it's good. From transferring it to other trophic levels, not a great thing. But we're still working to understand what some of their impacts might be. And then for our native lookalikes, I again have our pointed campyloma, which is much smaller than these snails will be and not nearly as bulbous. And this Leoplax, which again is textured, much smaller, not nearly as bulbous. So these guys have been fairly widely reported in Pennsylvania. You can see that it's essentially on all sides of the state. So there's a good chance that it's also located other locations as well, in the middle of the state. And this is our distribution for the Japanese mystery snail, where we have a lot more of it down south, but we do have instances up here in, say, Pymatuning Reservoir. And with that, I'm going to turn it over to my colleague, Sam Nutile, who's going to talk to you about how we look for snails.
All right, hello everybody. So I'm Sam Nutile, and I'm going to talk to you a little bit about how we search for snails, how to report and record what you find, and how to prevent the spread of invasive species to new habitats. So when looking for snails, one of the easiest ways to do this is to search the shallows of aquatic habitats. Now, not all of the species that Lynn talked about are going to be found in the shallows of aquatic habitats, but to get at snails in deeper areas, you usually need more specialized equipment, which can make sampling a little bit more challenging. So if you're out and you're looking for snails, one of the best things to do is just walk along the shoreline, either on land or wade through the water, and look for snails that may be present on things like coarse woody debris, so sticks, logs, stumps, other tree matter that might have fallen into the water. You might see some snails crawling around on this stuff or tucked into some of the nooks and crannies of those stumps and logs, or associated with things like vegetation, the mud just kind of crawling around in the dirt, or on rocks or other hard substrates in these shallow areas. When you're walking around, if you're looking for snails, you may be able to see them with your naked eye just in the water. So a good pair of polarized sunglasses can go a long way in terms of detecting snails in your aquatic habitat. It'll cut the glare off the surface and you may see the snails just crawling around down here. Or if you stumble across some woody debris in the habitat or vegetation, looking at the roots, or as you can see in this image here, these juvenile mystery snails are tucked away in kind of the pores and holes of these logs, crawling around and feeding in these places. So you can kind of just look and see where those snails are found. If you're a little bit more adventurous, one of the best ways to find snails is to actually take your hand and take the palm of your hand and just push it down onto the bottom and kind of pat around with your hand. You'll feel the snails under your hand as you do that. Now, this can be a little bit tricky because there are a lot of rock-shaped, snail-shaped rocks out there that we often confuse for snails. So you think you found one, you pull it up, it's a rock, which is a little bit disappointing. And other times you find all kinds of other interesting things when you're out searching in these aquatic areas. So keep your eyes open for all the kind of other cool wildlife that can be out there. So if you're out looking for snails in the shallows and you want to collect some data on what you're finding, one of the best ways to do that is to use what we call a quadrat, which is just kind of this square shaped structure here that we've made out of PVC, as you can see, that we use to look for snails within the defined area. So the quadrat that we use is made out of PVC that we've drilled some holes into. You can see here that allows it to sink in the water. Our quadrat is 1 meter by 1 meter square, and this is important that we know how big it is because it allows us to calculate density estimates of our snails in the habitat. So what we do if you want to do some quadrat sampling is you can build one of these for yourself. It doesn't necessarily need to be 1 meter by 1 meter. It can be kind of cumbersome to have one that big out with you in the field. If you want something smaller, that's fine. The only important thing is that you know how big it is. Because if you are going to try and calculate density, it's important to know how much area you're searching within your quadrat. But if you do make a quadrat, you go out into the field and you want to use it for sampling, you just head out to your aquatic habitat and just throw it out haphazardly out into the aquatic habitat, and where it lands, you go out and search for snails. So within the space of your quadrat, you look for snails in that habitat. When searching for snails, you can record what you find. So we bring out some waterproof paper here. It's called Write in the Rain paper. You can get it on Amazon pretty easily. And it works really well because it can get wet and it won't lose or won't kind of break down with that water. We record how many snails we find within each quadrat and what species we find. So here CC stands for Chinese mystery snails, and then here's our Japanese mystery snails. These are the abbreviations for their scientific names. And we use that information to calculate density, so how many snails we're finding per square meter. Within each of these quadrats, we also record some abiotic data about the habitat in which we find our snails. So we record the depth in centimeters here, so that's pretty easy to do. You can get yourself an extra piece of PVC and then mark out depth on it and just kind of use it as a stick. So we measure the depth in all 4 corners of our quadrat to see what depths our snails are occupying. And then we also usually take out some water quality meters to look at temperature, pH, conductivity, and other water quality characteristics to see what habitats our snails might be preferring. But if you don't have that equipment, depth is a really good thing to record, as well as the density of the snails in the area. If you do happen to find some snails out in your sampling, one of the best things you can do is record their presence with a picture. So when taking a picture of your snail, it's important to orient it the correct way so that people that may need to identify it later on can see what they need in order to make that identification correctly. If you find a snail, what you want to do is find flat surface, or a surface as flat as you can get that's well lit, and place the snail in that area. Having it well lit helps to avoid shadows so that we can see kind of the umbilicus or the shape of the aperture, or the pattern of the whorls and the shape of the spire clearly from the picture. You want to place the shell down with the aperture facing up so that we can see if there's an operculum in the shell, we can see the pattern on that operculum or the shape again of the aperture here, and then put something next to it that gives you some sort of reference for size. So if you have a ruler available, that's the best option, so we can actually see how tall the snail is or how wide it is based on the scale bar provided by the ruler. If you don't have access to a ruler, if you're out in the field or something like that, a coin is a really good option as well, because coins will all have a standard size that we can use to kind of guesstimate or estimate how big the shell is relative to that ruler. So taking a picture in this way is really important for identification. So the presence and arrangement of the operculum, the orientation and the shape of the whorls, as well as the size of the snail, can all be key features that can be used by experts to identify what species it is that you found during your sampling. If you do find snails and you take a picture of them, one of the things you can do with that information to help advance the management of these invasive species in aquatic habitats is report your findings to iMapInvasives. So if you go to the Pennsylvania iMapInvasive webpage, there's a couple different ways that you can submit a report of your findings to this organization. So one of the easiest ways is right here on the main page. You can submit a public report. If you click on this link, it'll bring you to this form where you can fill out your contact information, what you found and where, as well as upload a photo of the species that you've documented in your sampling. If you want to get a little bit more information out of this, if you're more interested in invasive species or other information, you can go back to the Pennsylvania iMapInvasives website and register to create an account. Once you've created an account, you'll get access to this database here that shows you information for invasive species across the United States, as well as parts of Canada, as we can see here, and also give you options to submit a report of your findings through this create record option here. Possibly the most feasible way by which you can create a report, especially if you spend a lot of time in the field and you maybe don't have the best memory like we do in terms of reporting what we find once we get back to the lab, is to use the iMapInvasives app. So this app can be downloaded on your phone through the Google Play Store or the Apple Store. And you can see here, you can add an observation right from the app here and take a photo in the app with the camera on your phone and upload it right here with the reporting information. So really easy, quick way to upload your findings when you're out in the field. And so reporting your information to iMapInvasives is really important because it helps with management of invasive species. By knowing what's present and where, we can document how a species might be spreading to new aquatic areas and do more to try and prevent that spread and manage what is present in these water bodies. Information that's also uploaded to iMapInvasives helps to provide a data set that can be used to understand the ecology of these invasive species in non-native habitats. So we benefit greatly from this information. We have lots of students that do lots of fun projects with these invasive species in these areas around northwestern Pennsylvania. So that goes a long way to helping out the science associated with studying invasive species in our aquatic habitats. And so while we benefit, I guess, from the science associated with studying invasive species, it's really not a good thing to have them spread. So when you're out sampling, one of the most important things you want to do is limit the spread of species from one habitat to another. And when focusing on how to stop the spread of invasive species, the most important thing you can do is when you're moving between sites, make sure you clean, drain, and dry your gear. So this involves inspecting and cleaning off your waders, your boots, your nets, your quadret, any equipment you took with you out into the field. Make sure you rinse it with the water at the site before leaving your sampling area. Remove any visible mud, plant matter, or animals that might be stuck to your stuff, because even though they're aquatic, they may be able to survive temporarily outside of water, stuck in your boots or in your waders or in a bucket. And when you go to a new site, they'll be released from that equipment and be present now in that new habitat. Also, if you collect anything while you're sampling, you want to try and dispose of all of it at the site from which you collected it. You do not want to transport specimens. So it's very tempting for people to remove invasive species from a habitat when they're out sampling. But as we saw from what Lynn presented, a lot of these species can be easily confused with natives. So while you may think you're throwing away an invasive species or killing an invasive species, removing it from a habitat, you may be actually unintentionally killing a native species, which is not a good thing. So we want to try and avoid taking anything from the habitat while out sampling. But finally, the best option when trying to prevent the spread of invasive species, the best thing you can do is to dry your equipment for 5 days or more. So a lot of these species, so the mystery snails, the other species with an operculum, can survive outside of water for a ton of time. But if you leave your equipment to dry for 5 days or more, most aquatic species will lose all the water they have retained after that time and die. So they won't be transported with your stuff when you go to a new area. If you don't have 5 days to wait for your equipment to dry, then you can try and wipe it dry with a towel is a good alternative to preventing that spread. So with that, I'm going to turn it over to Adam Simpson, who's going to tell you why we care about these species.
Hello, everybody. I'm Adam. So why are we focused on snails? Many of you probably don't think about snails all that much. And there's a reason for that. There's not a whole lot of information out there. We don't necessarily know what's out there, what levels, what their abundance is, and what effect they have on native ecosystems. So it's a large knowledge gap as far as invasive species are concerned. And when we try and categorize invasive species, or at least non-native species, a lot of it depends on what impacts we think they might have. So there is a difference between something that's simply non-native and introduced, as opposed to something we would term invasive. And the major difference there being that invasive species, we have some evidence or strong suggestion that they are causing some pretty severe impacts on the ecosystems that they've invaded. And like Lynn was talking about, many times we do kind of just guess that because they're here, because they didn't originate in this area, that there might be some sort of competition or displacement with native species that inhabit similar levels within the ecosystem. So other native snails that are similar sizes, they feed on similar things. We expect there to be some sort of competition between them, or at least the potential for this. But there are also other bigger, more indirect effects that maybe we can't foresee, or maybe they don't appear immediately, and those can be just as concerning, if not more concerning. And Lynn did talk a little bit about the potential for biomagnification or trophic transfer of contaminants. Could be things like heavy metals, organic pollutants, or even parasites that could be introduced to higher levels, recreationally important species like waterfowl and fish. And it can be difficult to predict these things, and that's why sampling is so important. We would expect, especially for these larger invasive snails, if you grow bigger and you live longer, you have much more time to accumulate these potential toxins. So what we did over the past several years, we started our sampling in 2019 and have continued it since then. Our major focus has been in Western Pennsylvania. So in 2019, our main focus was on Presque Isle State Park and the bays surrounding that area. As we continued throughout the next couple of years, so 2020, we also visited Lake Pleasant, Lake Canadota, as well as Pine Tuning Reservoir in a private property in Sagertown. In 2021, we also added Lake Arthur within Moraine State Park and the Allegheny Islands in Pittsburgh. And then in 2022, so this was this past summer, we added Edinburgh Lake, and we removed Allegheny Islands because we did not detect any when we went down there to sample. One thing you'll notice across the years, so these are the different locations we sampled within these different water bodies. You'll see that that effort has increased. So as we got better at figuring out ways and sampling techniques that worked, and we got more help, we were able to increase our sampling effort over the years, and I imagine this will probably continue. As far as the snails we found, so we were able to find, of course, the large charismatic mystery snail species, the Japanese and Chinese mystery snails in blue and green here, as well as the faucet snails in pink. As far as how to read this map, so I'm going to show some just general data from our last couple of years of sampling. You can see here we have indicators that suggest if we found data there, if we went sampling that area, did we find them or did we not find them in these specific water bodies? So we can see up here in Presque Isle, we were successful in finding both the faucet snail and the Chinese mystery snails, all for the years that we sampled. As we continued and expanded our research area here, you can see that we found Chinese mystery snails at many locations, and we had our first detection of Japanese mystery snails in 2020 in Pine Tuning Reservoir and Sagertown. And then as we continued in Lake Arthur, we did find both species of mystery snails. We did not find anything at Allegheny Islands, indicated by that tan circle there. We did receive some anecdotal evidence, I guess, of mystery snails being present there, but we were not able to find any. And then when we added Edinburgh Lake, we did find Chinese mystery snails, although they were not super abundant. And one thing I want to, I guess the main takeaway from this, you can see across the years, we added new sites and the number of sampling locations increased. And a lot of that was due to anecdotal evidence or just basic communication with people at conferences when we present, just talking to landowners. We got information, hey, I think I've seen these snails at this place. So we add that to the list, we go out, we sample there. So that goes a long way as far as helping us map out these misunderstood species throughout Pennsylvania. As far as our main focus moving forward, we are interested in generating population size estimates, as well as understanding what habitat factors predict the presence of these species elsewhere. And our major focus is on Presque Isle State Park, which is where this all began. This past summer, we did do some trial runs with a mark recapture study at 4 locations within Presque Isle. These are all relatively small water bodies with the exception of Thompson Bay. And these, of course, are areas that we've detected these species in the past. The way mark recapture works, it's relatively simple but powerful approach to estimate population size. You just need some way to mark a snail that you found, you release it, and then you have several successive days of resampling that area. You're trying to find snails that you've already captured as well as ones that you haven't marked previously. And we started with, we're mainly focusing on the mystery snails, at least at this point. We started with these nice little tags, and then we ended up finding out the first day of sampling, we found a lot of snails. So over 250 snails. We started running out of tags, needed a new approach. So we switched to these paint markers, essentially, that allowed us to give a color code to each snail that's unique to a specific site. And that paint lasted long enough for us to resample them over a period of a week. So what we did, we'd sample them the first day, we'd mark the ones we found, we'd return for 3 consecutive days. Each time we'd count the ones that were marked and the new ones that we had to mark and then release for several days. And ultimately, we found over 500 snails present within these 4 sites. And the way mark recapture works, it gives you information or can predict population size based on the proportion of snails that are recaptured versus ones that you find that were not previously marked. So if you have a situation where you're getting a bunch of recaptures in an area, the equation of the model is going to suggest that the population size is relatively small. Whereas if you keep marking them and you're not recapturing them, this is the implication is that it's a much larger population that you're dealing with. So these are our estimates from the different sites. You can see many of them are quite large. In brackets, we've got the confidence intervals of this estimate. Overall, our mark recapture model suggests that we have over 1,300 snails at these different sites in Presque Isle, although we will say this is probably a large or massive underestimate. When we went out and sampled, we noticed the snails weren't really moving around a whole lot, so we had potentially inflated number of recaptures in those areas, which we can account for with some more robust sampling techniques in future seasons. But the bottom line is there's quite a few snails there, and the abundance or density in some of these small water bodies is quite concerning. So things we're still working on, things we want to learn more about. This is one potential issue that we've been dealing with. We've done some genetics to try and distinguish between the different mystery snail species in particular. And what we've determined so far, at least from preliminary information, that we may have more than 2 mystery snail species. It may be as many as 4. These are all very closely related species to the point that many of them look identical. We have noticed some slight morphological differences, but we haven't necessarily associated these with the genetic ID. So they are appropriately named mystery snails. It's really hard to tell the difference between these species, especially if there's more than we think. Why that matters, it might not, but if they do have differences ecologically or life history, they reproduce differently, that might have different impacts and might change management practices. It's also harder for those outside of science to identify them and notify agencies what's there. We're also interested in looking at what eats these things. We have found evidence of predation, clusters of crushed or broken shells, which suggests maybe something like a raccoon eating them, but we haven't really seen a whole lot of this. We don't know to what extent birds are eating them. And especially if we're concerned with the transmission of parasites or other pollutants to higher trophic levels, it is important to understand exactly who is at risk here. And finally, part of our work looking with or trying to determine density and population estimates, we are keeping track of habitat qualities, habitat factors and parameters. So we're looking at the different type of vegetation that's there and other abiotic factors. And this gives us an idea of what environments, what habitats these snails prefer. And that can provide some information to help us predict where they might go or where they might already be. So Lynn brought up multiple times the fact that, they're reported in these counties. There's a lot of empty areas, empty gaps in between there. It is quite possible that they're already there. This information can help us figure out where to find them and where they might be moving to. And of course, we're Pennsylvania. We hold like a 75-mile shoreline on Lake Erie. There's obviously a lot more states involved with this. Mystery snails and other invasive aquatic snails have been reported throughout the Great Lakes. So we're just one small part of this, although we are an important part. So understanding what general environmental factors predict the presence of these species can be important for management on a much larger scale. So what can we do? At this point, obviously it's December. There's not going to be a whole lot of snails out there. We notice that they typically move into the shallows in early June or late May. So there's a lot of time between now and then. One thing that can help is the Pennsylvania iMapInvasives website, which we've already showed you a little bit about, but the resources tab in particular I found quite helpful, especially getting background information about these different species, because we did introduce several. The gallery of invaders is good as far as getting looking at pictures and getting basic information about what these species do and where they are. And the training archives is a good resource to look at examples of how we sample for invasive species. So we don't have a snail sampling video up yet, but if there's enough interest, we might be able to put something together. But there's a lot of helpful information on how to appropriately sample in a way that you can help people that are using this information to ask bigger questions. We also wrote an article through the newsletter for iMapInvasives last year that you guys can take a look at. We kind of describe what a day in the life of a snail sampling team is. So you get to see a little, bunch of pictures of us sampling, how that process works and so on. So that is available through iMapInvasives. And as far as things you can do, spreading the word's big. Not that you're going to just be talking about snails all the time, but there might be situations where something comes up. Maybe your friend is looking at buying snails to clean their pond or their tank or something. A lot of those snails you purchase online are not from Pennsylvania. Surprise. We actually found a vendor online on Amazon that you can buy mystery snails, and they're advertised as pond cleaning snails. So no surprise, they're popping up in like golf course ponds and things like that. So there might be a situation where you can share your knowledge of invasive snails. Report what you find. So when we finally get to the time of year where you're outside doing stuff, they're out there. You've probably seen them and not realized it. If you have time, especially if you have the app on your phone, it's easy to just log in sighting. Like Sam mentioned, it's probably a little bit more convenient to set up an account and log it in through there, but you can submit a public report without being, without having an account set up. So that's very helpful. It might introduce or let us know about a site that we haven't sampled before that we need to visit the next year. And finally, lend a hand. It's possible next summer, we usually have an army of undergraduates that help us, but we might need help sampling different sites next year, next summer. So if you're interested, let one of us know or let Amy know, and we can keep your name in mind for future sampling events. You can always use the help, and there's a lot of snails out there. We'd like to acknowledge several things, several sources. Of course, Penn State has supported us. We've received financial support and other help from Pennsylvania Sea Grant. We've got permission to sample a variety of different places across the state from Fish and Boat and other park managers. And we've had a long list of people that have helped us sample over the years to acquire our data. That we will take any questions from you guys.
Okay, awesome. That was a great presentation. Thank you, all three of you. I really enjoyed that, and I'm certain that our audience really enjoyed all that great information. We did have some questions that started coming through, so I'll go ahead and ask those, and then I actually have some questions myself also. So the first question is from Nick. He is asking, is there a visual way to identify a calcareous operculum like the faucet snails, or can it only be identified by how hard it is?
It's going to appear more opaque than the corneous ones are, because the corneous ones, I should have brought some in here. but they're really thin. They're often flexible, like potato chip thin, while the other ones are going to appear much thicker.
Okay, thank you. Nick also had another question. He's asking, what is the habitat preference for the big-eared radix?
They tend to be in slower moving, still bodies of water, but they are quite adaptable. So there are some areas where they've invaded, they used to be just restricted to more shallows, I believe, and then they've adapted to inhabit different parts of, I can't remember the example water body I'm thinking of right now, but they can be quite adaptable. Yeah.
Okay, great, thank you. We have a question from Jeannie. She's asking, why aren't the captured snails destroyed?
So the ones that we capture are destroyed. So everything we collect during our sampling is brought back and we clean the shells to photograph them, and then we freeze them to euthanize them. And we dissect them for metal analysis. So we look at the composition of metals in their shells and soft tissues. So everything that we collect that we can properly identify, we do destroy.
Okay, thank you. We have a question from Timothy. He's asking, Asian mystery snails look large enough to eat. Cooking them would kill parasites, but is the metal concentration dangerous for human ingestion?
That's a good question.
Yeah.
If you're ever going to consider eating wild-caught snails, it's a good idea to keep them in a tank for a good period of time. Just they purge a lot of those parasites. We haven't identified the parasites yet, but the ones that we looked at, they're full of like liver flukes, and there's a lot of stuff in there. And sometimes the encysted parasites aren't destroyed by cooking either. So that's a little bit of a risk. But as far as metal concentrations, I believe barium was the highest that we found. With
Yeah,
other
and copper.
tissues. And
There's
copper.
a good amount of copper in there too.
Most of the other ones that we're concerned about, things like mercury, lead, cadmium, are quite low concentrations. If you're feasting on a bunch of them, maybe it's more of a concern, but our concern's largely not human-based. I think that the main thing might be if fish or birds are eating a bunch of them.
Okay, so not something that we recommend.
I would find other snails maybe.
Okay, good to know. We had another question from Jeannie. She's asking, are bladder snails a huge problem in Pennsylvania? Physa acuta.
They're native,
So not a problem.
not a problem.
Question from Andy. It was mentioned that snails move into the shallows in June and July. Where are they in the winter? Deeper water or buried?
Both. Depends on the species, but they do both. Yeah, a lot of them bury, a lot of them go deeper where it's not going to freeze.
Okay. Another question from Timothy. Do you ever encourage surveyors to submit voucher specimens for deposit in a museum? Carnegie Museum would accept reasonable numbers of vouchers.
Yeah. Yeah, I mean, and Carnegie has like one of the foremost snail experts out there, Tim Pierce, who could definitely ID things accurately too.
Yeah, that's who's asking the question.
Oh, of course.
Okay, very good. Very good. And another question from Tim Pierce. You recommended verifying ID by an expert. By expert, do you mean you guys or someone else?
You know, I was really thinking you, Tim. But yeah, you probably know more than we do. We can certainly try, but we probably defer to you anyway.
Very good. We had a question from Mark. Are there trapping or baiting methods that are effective for snails?
Not that I am aware of. I mean, food would be a good option, but they're not all attracted to the same food thing. Plus, a number can like graze and filter feed or like pellet feed. I'm aware of. So if someone knows, please let me know. So
Great, thank you. We had a question from Sean. Do you think the map turtles on Presque Isle feed on the mystery snails given their dietary preference for aquatic mollusks?
it's possible. I would bet that at a certain extent, the mystery snails are too big for the turtles to eat, but they could probably eat the smaller ones. Yeah, and they're gonna be in slow, like not moving water. So yeah, definitely possible.
Okay, great. We had a question. Is there a handy printed identification chart that's available?
Printed. There's a book from '92, I think, that's like, it's more of a report on freshwater snails of New York. I can't remember the last name. It's like Jokenin or something is the last name that's printed. Most of the snail ID things are drawn rather than pictures.
Dichotomous.
Yeah, there is an online dichotomous key. Freshwater Gastropods of North America is the website, I believe, and they have regional dichotomous keys. It's dichotomous because it's not quite dichotomous, but close enough. But they do, it's pretty good, and it goes to actual pictures. It is online and not in print though, but it is very nice.
Okay, and maybe what we can do in my follow-up emails, we can include a link to that. So if you guys just want to send that to me, make sure I'm sending out the right thing, then we can pass that on to everybody. Okay, very good. And then Tim Pierce commented that he is actually a land snail expert, not completely reliable with freshwater snails. Okay, and then Jeannie commented, my information on bladder snails stated they were native to France and invasive to the United States. So they are now considered native to Pennsylvania, she's asking.
As far as I knew, they were native to here, invasive in Europe, but I will double check on that though. I could be having my locations backwards.
Okay, and I see your contact information is still on the screen. So if Jeannie would like to maybe reach out to you guys for more info,
Yeah, certainly.
that can happen. Okay, Jeannie said thank you. Edwina is asking, these snails that you presented today are aquatic. Is there a concern about invasive terrestrial snails?
Glad that Tim's here. The only one the ones I know of are like the garden snails, like the black and yellow striped. Then there's the giant African land snails. Those are more south.
Yeah, it's Florida.
Yeah, I don't know too much else about up here for land snails. So I'm gonna have to defer to Tim.
Okay, very good. We had a question from someone asking, what can you tell us in the eastern part of Pennsylvania about New England mud snails?
England mud snails.
I'm wondering if that is New Zealand mud snails.
Might be New Zealand. Yeah. There is someone that works in another Penn State campus that studies New Zealand mud snails in the eastern part of Pennsylvania. So I'm not as familiar with what's going on over there. If they're hyper abundant, I believe you have the parthenogenic line over there. I'm not sure, like control is incredibly difficult for any of these because any molluscicide that you would apply would just kill off everything, which is not good. So yeah, I'm afraid my knowledge of what's happening on the eastern side of the state is not good, but I can find the name of the other Penn State faculty member and send them their way or send publications.
Okay, that would be
We were looking for a New Zealand mud snail when we started sampling at Presque Isle, because there is a report in Erie County, but that report, that was sampling that was done really deep in the lake. We haven't found anything like actually in the shallows yet. Doesn't mean they're not there, but we haven't found them yet.
okay. Thank you. Let's see, Tim Pierce commented, pretty sure Rob Dillon of the Freshwater Gastropods of North America website is the one who thinks Physa acuta is a European snail.
Okay.
Okay, and Pete is saying NatureServe now has a Physa acuta listed as native in some states and exotic in others, probably differences of opinion between states. I think there is uncertainty about whether there are multiple species and where each started.
Physa, big mess.
And let's see, Sean commented it's Ed Levery from Penn State Altoona who is working on New Zealand mud snails.
Okay,
That's right.
excellent.
Thank you, Sean. And Cheryl just commented, awesome presentation. And I would second that. That was also, I also felt that was a really great presentation. I had two questions. Do you know, is there any biocontrols that are available or being researched to try to manage some of these invasive snails?
Not that I'm aware.
No, I mean, these guys have kind of been under the radar as far as control, I think largely because they're not, at least many of them don't cause any catastrophic effects that we've seen so far. They're just kind of there, and the effects that they've had are just not quite as apparent because not a lot of people look at snails, unfortunately. But yeah, I don't know of a track record for biological control.
I've read in other parts of the world, there's where parasite infection is more of a problem with snails and transmission through intermediate hosts. They try and bolster crayfish populations to act as predators to control snail populations in ponds and things like that. I don't know of anything done in the US.
Okay, great. Thank
Especially
you.
for snails that big, for like mystery snails.
Right, yeah. And then my other question for you, do you guys have any particular procedures after you're finished sampling to make sure that you're parasite free and you don't have to worry about spreading anything that you're bringing home on you, like with your arms and hands in the water? Do you have any specific cleaning protocols that you guys follow?
I think we clean ourselves similar to the gear. I mean, not that we dry for 5 days. That we wash everything off. A lot of the parasites too, you can't get without, at least for human health, it's you have to ingest in order for it to have negative consequences. So, I mean, we don't lick our arms. We wash stuff off with soap, clothes and stuff, sanitize.
Very good, very good. And we got another comment from Zachary saying, I agree, very cool presentation. So I think it was well received. That was everything as far as the questions that came through. So I just want to say thanks again for your time and your expertise. And I'll be sending out a recording for this later on. So thank you all and have a wonderful day.
Yeah, thanks for coming.
Thanks for coming.
Thanks.